Hello, everybody. I'll share my screen here. Just a minute. Let's see. Sorry. It's a little tricky when there's so much going on the screen. Here we go. Now I'm sharing my screen. And hello, everybody. I'm honored to be able to give you a 20-minute lead in to today's session. And my goal here is to really zoom out in thinking and offer some ideas to you. I know this conference is about honeybees, but I've taken the liberty of titling this talk to be all the bees. So I'm hoping to broaden our thinking a little bit. I'll begin with honeybees though. And I want to begin by pointing out that honeybee colonies are very amazing. We consider them an example of a superorganism, which means the colony is the animal and it's comprised of thousands of animals. And each of those individual bees cannot live without its colony. So honeybees and ants and termites and some of the wasps are considered social organisms, and they have no central authority, no government. There is a queen, she's the mother, the egg layer, but there's nobody giving instructions. And when we think about honeybee colonies, it's very important to understand that imposing our human thoughts and views on the colony or things we think they need is not really appropriate sometimes. It's more appropriate to think like a bee and to understand what they need and where they're coming from. And so I'll just I'll just drop that little thought as I continue. Honeybees are both wild and managed. Some of them can still live fine on their own as feral colonies in a tree cavity. Most of the honeybee colonies in the United States are managed. And we keep them in these boxes, of course, where they're known for collecting more honey than they can use. Honey, of course, is floral nectar that they store in cells and then convert into honey. They also collect pollen, as shown in this lower left-hand photograph of the different colored things stored in the cell. Those are pollens from different floral sources. That is their protein source. I'll come back around to that. There is a queen shown here in this middle bottom photograph with a dot of paint on her, surrounded by her female workers. It's a female-based society. And of course, we can keep them in these boxes where we can manage them and lift out cross sections, the frames of this animal to look deep inside its living being and its organization. We can keep them in our backyards as a hobby. We can keep them in mass, as shown in the lower right photo of a holding yard outside the almond orchards or near the almond orchards in California, where thousands of colonies are trucked in and located in one location, getting the colonies ready to pollinate the almonds at the end of February or whenever they bloom. The next few slides are information that probably everybody is aware of, that honeybee health is in decline. Honeybees populations are not going extinct. They're not at risk, but their health is problematic. The health of a honeybee colony is suffering. And we call them maybe the 3 or 4 P's of honeybee health decline: pathogens and parasites, pesticides, and poor nutrition. And these factors interact in ways, sometimes some more than others, but in various combinations, to often put colonies in a downward spiral of poor health. So their main, arguably, but I would argue, their main pathogen and parasite problems are this Varroa destructor, which is a parasite, a mite that feeds on bee tissue. It picks up viruses that are inherent to honeybees, or sometimes other insects also. And as the mite feeds on the bee, it picks up some of these viruses. Some of them they can replicate, and then they transmit these viruses. The mite enters cells containing developing pupae, shown here on the right, the developing bees, and they also feed on the pupae and reproduce there. They greatly weaken the bee colony and reduce their lifespan. And of course, the viruses, just like we all know with COVID viruses, the different viruses that honeybee colonies have can also take them down. When they're outside foraging, they encounter a number of pesticides. If you collect the pollen from the back of their legs, as shown, these orange blobs that are on the hind legs of these honeybees returning to their colony, if you collect a sample of those and do pesticide residue analysis of them, you'll find in varying degrees, sometimes at very low detection levels, sometimes at very high detection levels, insecticides from all classes of insecticides, fungicides, herbicides, and although we don't know what they all are, adjuvants that are put in pesticide mixes. And as we study the effects of the exposure of these individual pesticides on bees, we now know that those effects can be synergistic, not just additive. In other words, what the effect of a pyrethroid might have on a bee, plus fungicide, for example, may not be the sum of the parts. It may be more than the sum of the parts to be more harmful. Not always, but can be. So as research reveals the effect of pesticides, we know that pesticide exposure to bees that are already riddled with varroa mites and viruses are much more toxic or hard on the bees than the effects of pesticides might be alone. And then as bees forage in their environment, they may encounter situations like this one in Minnesota, which is where I live, that is predominantly corn and soybeans from roadside to ditch. Due to our industrial agricultural practice and even surrounding our homes and farmlands, which are all lawns, which is another monocrop, bees may struggle to find the flowers that provide good nutrition for them, the nectar and pollen that they really need for survival. But it's not just about honeybees. It turns out it's about all of our bees. In North America, we have over 4,000 species of bees. Most of them are native bees. Honeybees are, of course, not native, and all of them contribute pollination services and therefore to our food security. Most of our native bees, are solitary. They do not live in huge social colonies. They live on their own in the ground or in stems, as I'll show you, and they have varying morphologies and tongue lengths, which allow them to get the nectar and pollen from different kinds of flowers. There's a huge biological diversity of bees, and it is extremely important that when we think about honeybees, we do not disregard all of our native bees. Our wild bees, our native bees, are facing similar problems of our honeybees. They're also exposed to pesticides and poor nutrition, often from lack of flowers, and pathogens and parasites. I'll come back to that. But I would add another factor for wild bees, and that is sometimes a lack of nesting sites. And this can happen in our agricultural areas where we till the soil and we disrupt their nesting sites. It can happen in our urban environments if we overmulch our gardens. So some of these bees, and the majority of them, nest in the ground. And so they're on their own. One individual female bee makes a hole in the ground, puts some pollen down in there, lays an egg on it, seals off that little section into a cell, and then maybe brings in more pollen and a few more eggs. And then those eggs develop into a bee, but it often takes the winter. It takes months, 9 months or so, for these bees to develop. So they spend the majority of their life in the ground. And when we till the ground, or we make it so that when they emerge finally the next spring from the ground and they can't make their way out through so much mulch or something, for example, or pavement, then their nesting sites are disrupted. The other third live in tunnels or stems. And so the same kind of biology, most of them are solitary. The female would bore into a stem that's usually hollow or very pithy anyway, put some pollen, lay an egg, make a partition in the stem, and continue leave the nest completely, and her young develop on their own over the winter. So if we take down the stems in our garden, if we take down the stubble, the corn stubble, for example, in our fields, we're disrupting these nests. The pathogens and the parasites that these wild bees face may be different from those of honeybees, but as we're learning, some of the problem may come from honeybee colonies themselves, as this photo over here in the lower left-hand corner. And this is kind of disturbing news, but one that I think that we should all pay attention to. And that's the risk of pathogen spillover. And it works kind of like this. So if you have some honeybee colonies in an area and they're full of disease, for example, they have a lot of virus or a disease called nosema, and they visit flowers in the area, and let's say there's not that many flowers, they visit a flower and inadvertently deposit or leave some of that pathogen right on the petals of the flower, right on the flower itself, and then leave. And then step two would be one of our native bees, here shown as a bumblebee, comes into the same flower to forage, to pick up her pollen and nectar herself. And then she may end up picking up some of that virus or that other pathogen in step three in what we call spillover. The risk of this is still being studied. We know it can happen. We don't know the extent of it, and we don't know the consequences of it to our native bee populations. For example, even though we can detect some of these pathogens in our native bees that we know came from a honeybee, we don't know if it actually affects the lifetime fitness or makes our native bees sick. The detection of the pathogen does not necessarily mean it affects their overall health and lifetime fitness. However, it seems logical to assume that if there are lots of colonies in one area and very, very few flowers, the honeybees that come from these colonies that visit the few flowers increase the risk of pathogen spillover to our other bees. So of course I've been, and my background shows, this is my, I should say this in the background is my front yard here in Minnesota, although I'm not in my front yard right now. That's why I've been saying for years that it's very, very important to provide more floral nutrition for all of our bees, because good nutrition helps bees stay healthy in exactly the same way as good nutrition helps us and all of our farm animals stay healthy. So the pollen that bees collect from flowers provides their protein and lipids, and the nectar, of course, provides their carbohydrates. But in addition, and importantly, pollen and nectar contain these plant chemistries or phytocompounds that we know now they're like value-added compounds, antioxidants, etc., that bolsters bees' immune systems and help them detoxify pesticides and other toxins. So some of these phytochemistries can stimulate genes to make enzymes to produce the P450 detoxification system in bees, or they turn on genes that make proteins that help the bees' immune systems. So it's not just the nectar and pollen, it's the good nutrition that they're getting from the flowers themselves, from these chemistries. And so we need more flowers for bees to detox and restore their immune function, and also to reduce the risk of competing for the same flowers and pathogen spillover. That's the way out of that potential problem, is to make sure there's sufficient flowers for everybody. And the way to do this, of course, is through planting more pollinator habitat. And if you zoom out from that, simply planting pollinator habitat provides multiple and stacked benefits. It is also wildlife and bird habitat. And then if you look in the ground, if you keep the pollinator habitat in the ground, it sequesters carbon, which moving forward and finally is critical to our survival on this planet. The long root structure in many of the perennial plants we put in for pollinator habitat, and even if we're putting in cover crops, The roots prevent soil erosion, which then filter pollutants into waterways so that we can improve our water quality. Zooming out, a little bit of pollinator habitat goes a long way toward mitigating climate change, improving our water quality, and our soil health, which is finally something that now we're all thinking about again. Our soils are depleted now from overmanagement and our farming practices, unfortunately, and we're returning to, I would say, thinking about more regenerative agricultural practices, where we're concerned about soil health and how we can keep plants in the ground, how we can do crop rotations, and how we can improve the soil health, which would then improve plant health, and it just goes on from there. I'm proud that the Almond Board is considering cover crops like this canola that's planted here between the rows. In this case, I took this right from the website. It's planted and showing how it helps compacted soils between the rows. But of course, canola is a bee plant that would really help bees, honeybees, and increase the population of native bees in the almond orchards, which unfortunately there aren't many native bees around the almond orchards anymore. And I hope you'll hear more about this in the talks that follow mine. I just wanted to drop that in. So as our soils are depleted, so is bee health. As the soils are overmanaged, so are honeybee colonies. And what are the ways out of this thing? And how can we possibly use new technologies? Well, I'm just going to offer some ideas that came to my mind. Let's focus on the colonies that survive and that thrive. We know from national surveys that at least a third, sometimes 40% of all of our honeybee colonies die off every year. And there's about another third that just kind of eke by. But there's a certain percent that thrive, that do really well. And we don't tend to focus on those. We're concentrating on the deaths, the mortality, the causes of mortality. Let's flip our thinking a little bit and think about that top third. That is our breeding and selection material. There's a reason that some colonies can thrive despite the pesticide or pathogen varroa pressure, despite nutritional challenges. There are colonies that do really well. Let's learn more about them. So how can we do that? Through our technologies, can we focus on honeybee colonies that consistently have low mite and viral loads? Can we follow those colonies longitudinally over time? Can we track those colonies? Do they always have or consistently have low mite and virus loads? Can we use those as our breeder colonies? Can we find the colonies, that top third or however, whatever the percentages that have really good nutrition? What are the markers in a honeybee colony for good nutrition? I've put up a couple, but I know there's lots of research ongoing right now. If they have lots of incoming pollen, that protein and that lipid, that's an indicator of good nutrition. If they have VG, which is shorthand for vitellogenin, a very important storage protein for insects in general and honeybee colonies, that's an indicator of nutritional health. And there are others. What other ways can we track nutritional health of our colonies? How can we develop rapid and inexpensive detection of pesticide contamination in their wax, where pesticides tend to accumulate, and in their food stores, in their stored pollen in particular, and sometimes in their stored nectar and honey? How can we give beekeepers rapid and inexpensive results of pesticide contamination? We're looking at a lot of new management apps. A lot of them are great, and there's a number of them. Let's use those to track the thriving colonies. We tend to go in an apiary, sample colonies, find colonies that are over a treatment threshold for varroa mites, for example. There's a certain number of colonies that have over 3 mites per 100 bees, for example, and we treat the whole yard. Well, we can do that, but at the same time, how can we find the colonies that had way under the treatment threshold? As I said before, how can we track those thriving colonies. I skipped a slide. There we go. So zooming back out, when we think about bee health, it's important to focus on increasing the diversity around us, the biodiversity, the pollinator diversity, agricultural diversity, even diversity within our urban landscapes. Think about our lawns and that monocrop. And because all of that, when we have landscape diversity and pollinator diversity, we have nutritional diversity, many foods and vegetables that are good for us. And I think the most important and most tasty fruits and vegetables and coffee that are bee pollinated, that are so important for all of our health. So as this day moves forward, I encourage everybody to keep their eye on honeybees, but also to zoom out, consider all of our bees, all of our agricultural practices, how we can regenerate our soil and our agricultural practices, and how we can increase biodiversity. And I'll stop there, and I'll stop sharing and say thank you very much.